Bismillahirrahmanirrahim. In the name of Allah, the most merciful and the most beneficent. So today we will discuss about multiple sequence alignment, also known as MSA. So before that, we just study about sequence alignment and local alignment, global alignment. So today we will jump on multiple sequence alignment. So the first we studied about pairwise alignment, then now it is multiple sequence alignment. Aligning or alignment of two different sequences representing in two different metrics is known as pairwise alignment. However, if we have to align more than two sequences, or three, four, or five, or any number of sequences, that is known as multiple sequence alignment. And every sequence has one matrix in a row. So here we have three sequences: AT, GCG, AT, ACGTA. So here we have some gaps and we have three different sequences, and we are going to align those as a multiple sequence alignment. So every amino acid has a specific score that we usually use for gap penalties, and we calculate it. And we already discussed in detail all these in our previous lecture of global and local alignment. So what is multiple sequence alignment? It is specifically a model to align more than two sequences and to get a specific result for motif, for domain searching, for motif searching, or for three-dimensional structure prediction, and many other similar type of processes and experiments. Multiple sequence alignments also indicates relationship between the residues of different sequences, between the similarity, or between indicates the relationship of similarity between different sequences, their physiochemical properties, and many other things. Multiple sequence alignment also reveals similarity and dissimilarities from the sequences we use for the multiple sequence alignment. So why we need multiple sequence alignment? Multiple sequence alignment is central to many bioinformatics applications. In fact, alignment, blast, sequence alignment, and multiple sequence alignment, these are the base of bioinformatics, because we need so many similar analysis for our experiments in with the help of multiple sequence alignment. If we want to generate phylogenetic trees, or we just want to analyze adaptive evolution or behavioral study, we have to perform multiple sequence alignment. If we want to search motif or functional domains, diagrams, or different domains of proteins, we have to perform multiple sequence alignment. If we want to search a specific pattern in different sequences, we again have to jump to multiple sequence alignment. Similarly, if we want to predict the structure of three-dimensional structure of proteins, we again have to perform multiple sequence alignment or pairwise sequence alignment to just grab the target sequence for template sequence for the target sequence. So aligning three sequences is like the same strategy aligning two sequences. In two sequences, we align sequence A with sequence B. In three, if, there are different methods. First, we have to align A and B. Then we have align A and B with C, or sometimes simultaneously all three sequences. So use a three-dimensional Manhattan cube that is here. You can see one side is the source with this red ball, and then the last here is the sink. So we have to move from source to sink. If we we have to move to from source to sink if we want to analyze these three sequences or more than three sequences. For global alignment, we have to move from this point to this point, and we have to check all the possible outcomes, and we have to give a specific score to every gap, so we can calculate the maximum score for multiple sequence alignment of those specific sequences. So here you can see, if we have two-dimensional, then we have to move from here to here. Like here, we just perform back tracing in our previous experiments. So similarly, if we want to move from if we have more than two sequences and we have to move to three dimension, then we have to use this box, and we have to move from this point to this point. So here we have a profile representation of multiple sequence alignment. Suppose we have these sequences, 1, 2, 3, 4, 5 sequences, and we have to align those sequences. Specifically, if we will give, with here we have 5 sequences, and if I will give, we will give 0.2 to each this point, like 0.2, 0.2, 0.2, 0.2, and 0.2. If we add 5 times this 0.2, that will be 1. So that specifically give one specific number to this first column. So we have to give a specific number to each position. So on gap, we have some values deduction, then on the mismatch. So we can calculate it easily. So here we have this one, like here we have A. In this one, there is no A, so we have 0 here. However, we have 3C. So with the help of 0.2, we have 0.6. No G in the first column. Then T has 0.2, and the gap is also 0.2. So the first column we have 0.6, 0.2, and 0.2. Similarly, in the second one, all are A. So with the help of 0.2, we have score as 1. Here we also have score 1. Then we have 0.2, and then we have a 0.8 as a gap penalty. So seems that the gap will be more accurate here because we have to calculate the maximum score, and 0.2 is smaller and 0.8 is the larger one. So the gap will be here. Similarly with the C, T, A, and now we have 2C as a 0.4 and T as 0.6. Then we have C with 1 and A with the 0.8 and G with 0.2. Again, we have a gap here with 0.2 and 0.8, and then we have 1. So if this is a very small example for understanding. However, when we have to do more than 3, 4, 5, 10, or more than 100 amino acid in a sequence, then it will be a long calculation. So different software just perform these types of calculations. This is for our understanding that how this actually calculates. So these alignment, we can do alignments like these. So Cluster W is considered as one of the most famous software for multiple sequence alignment, most cited tool. Nowadays, it is also known as Clustal Omega. So there are 3 steps for performing Cluster W, that is construct pairwise alignment first, then build a guide tree, and then progressive alignment guided by the tree. So here we have a Cluster W algorithm. Yes. So here first we have 2 sequences. Here it is represented as blue, dark blue, and light blue. Then we have this alignment as here is some gap, and this is the first alignment. There is 2 more sequences. In actual, these are 4 sequences, and here we have the alignment of these 2 sequences. After that, we just align these 2 sequences with these 2 sequences, and now we have this 4 sequences with the gaps. This is dynamic programming using a substitute matrix that we just move back with the help of trace packing. We already performed this in our previous lectures, that how dynamic programming works and how we have to calculate it with different gap penalty, mismatch, and matches, and now how we have to use the trace back option. So similarly, here we have, if we have these 5 amino acids in one sequence, 1, 2, 3, 4 sequences. First, these align 2 sequences and these 2 sequences. Now we have this result, and then we align it separately for multiple sequence alignment. So Cluster W actually works on this algorithm. If there are 4 sequences, Cluster W will align 1, 2 first, and then 3, 4. After that, Cluster W will align the result of 1, 2 with the result of 3 and 4. That it will be more similar and will be more accurate. After that, we have progressive alignment, that has a specific scoring scheme. So that scoring scheme is arguable by the most influential component of the progressive algorithm. Here we have 2 types of algorithms. First one is matrix based algorithm, and second one is consistency based scheme or consistency based algorithm. So Cluster W, Muscle, and K-align softwares are based on matrix-based alignment algorithms. As we already discussed, that there is a matrix, that matrix has specific values like gap penalty, match, mismatch. So Cluster W, Muscle, and K-align used that approach of matrix-based algorithm to solve the multiple sequence alignment, and use a substitution matrix to assess the cost of matching 2 symbols or 2 profiled columns, and also give some specific deduction of numbers to the gap penalty or mismatch. Similarly, we have another algorithm or scheme that is known as consistency based scheme, and T coffee and die-align softwares are just based on that consistency based schemes, and compile a collection of pairwise global and local alignments to use this collection as a position specific substitution matrix. So why we have to align the sequences? because lots of sequences are available and there are still number of sequences that is unknown to us or to this world for their structure, for their behavior, for their properties. So we align the sequences, unknown sequences, with the known sequences to get that specific information for the unknown sequences. If they are similar, then we can consider that those are from the same class, same family, have same behavior. Or sometimes they have similar function, because similar structures has similar functions. We can also hunt out the conserved part, conserved motive, conserved domains, diagrams, more, and these types of useful information. If one of them has known structure and function, then alignment of other might yield insight about how the structure and function works. Similar motive content might hint to similar functions. So principles of sequence alignment, there are some principles, some rules we have to follow during the performance of this experiment of multiple sequence alignment. Alignment can be helpful to elucidate the homology modeling between homology and similarity between two sequences or more than two sequences. Similarity is descriptive term that explains the degree of match between two sequences, that how much percent and how much or how extent these two sequences has similarities. Sequence similarity does not always imply on a common function. However, conserved function does not always imply similarity at the sequence level. Convergent evolution we can also study with the help of sequence alignment. Sequences are highly similar in the past, and if they are similar, then their functions will be similar, their behavior will be most probably similar, and they will be not homologous to each other. So what is pairwise alignment? Here we have two different alignment methods, that is global alignment and local alignment. We studied these two alignments, global alignment and local alignment, in detail in our previous lectures. However, here we have just a look again to just recall what in actual global alignment and local alignment. The alignment is stretched over the entire sequence length to include as many matching amino acids as possible, up to and including the sequence ends. Vertical bars between the sequences indicate the presence of identical amino acids. For example, we have a very famous global alignment algorithm that is known as Needleman-Wunsch algorithm. And in local alignment, we have Smith-Waterman algorithm that is most famous. And in local alignment, we just have to take a look to the specific conserved portions to a small number of amino acids from a big sequence. So for those we just use local alignment. If we need conserved part, if we need motifs or domains, we just move towards local alignment. So these are in actual pairwise alignments. Pairwise sequence alignment versus multiple sequence alignment. In pairwise sequence alignment, we usually use two sequences. However, in multiple sequence alignment, more than two sequences are used for the alignment. Pairwise sequence alignment is used to identify specific similarity or specific region between specific region of similarity in two or more than two sequences. Sometimes that may indicate the functional, structural, or evolutionary relationship between that two specific biological sequences that are used for pairwise sequence alignment. Here we have an example. Q, and this you can see that where we have a match, there is a bar, and when there is a no match, there is no bar. Say, and in the meanwhile, if we are just going to study multiple sequence alignment, the alignment will be among more than two sequences. And what is the similarity between these more than these two sequences we are going to use? What are the length? What are the behavior? How this protein behaves? What will be the structure of these proteins, and what will be the homology and similarity between these two proteins. In actual, also we can study the evolutionary relationship between these sequences that are more than two, as for multiple sequence alignment. Here we have an example of that. Here we have three sequences. However, in the pairwise sequence alignment, we have two sequences. Different computational approaches to perform a multiple sequence alignment. The dynamic programming approach, we studied it in detail in our previous lectures, progressive alignment method, and iterative refinement method. In dynamic programming approach, we have to find the optimal alignment between the two sequences, that how these two sequences optimally aligned or suitably aligned, what will be the maximum similarity between these two sequences. For the alignment of protein sequences or DNA sequences, we specifically use some scoring metrics, but in practice often simply assign a positive score to the match and negative score or zero to the mismatch or gap penalty. For more than a few sequences, exact algorithms become computationally impractical. This is why this method is not widely used nowadays. However, it was a very good method still for some type of experiments. The runtime increases exponentially with the number of sequences that we want to align. In progressive alignment method, we already studied dynamic alignment methods in detail in previous lectures. If someone wants to learn more about that, he or she can consult those lectures. Progressive alignment method, the most widely used approach for multiple sequence alignment nowadays. Heuristic algorithms are used for multiple sequence alignment, and it is considered as the most fast and accurate method or algorithm for multiple sequence alignment. Progressive alignment builds up to a final multiple sequence alignment by combining pairwise alignment beginning with the most similar pair and progressing the most distantly related. So T coffee, cluster W, and pipe up are considered as most reliable softwares for that are based on progressive alignment method. So here we will study the progressive alignment method in more detail, so we can understand specifically how this method works. All progressive alignment methods require two different types of stages, two stages in actual. In first stage, the relationship between the sequences are represented as a tree that is also known as guide tree. Here we can see that here's step one. We have sequence one, then we have sequence two, and there is, you can say, G with the A. However, there is no similarity. But in the first step, we just generate a tree that is known as a guide tree that will guide us for the result. And second step in of multiple sequence alignment based on progressive alignment method, we have to add the sequences sequentially to growing multiple sequence alignment according to that guide tree. So here we can see in the first step we have two sequences, and here we have some gaps. However, in second step, we add the third sequence. In third step, we add the fourth sequence. And now again and again, we will add the sequence. One by one, we will add the sequence and we will align the sequence. Suppose in sequence one, we have 4 amino acids, and in sequence two, we have 3 amino acids. First, these two will be aligned. After that, we have in sequence three, 5 amino acids. Then these 5 will be aligned accordingly, first and second sequences. Then the fourth one will come, and that fourth one will align according to first, second, and third sequence. So it all depends on that guide tree in actual that will lead to the multiple sequence alignment. A popular heuristic algorithm is Clustal by Higgins and Short in 1988, and still it is widely used algorithm nowadays. Clustal makes a global multiple alignment using a progressive alignment approach. So here we can see that I already tried to explain in the last slide that first they will align sequence 1 and sequence 2, then sequence 3 with 1 and 2, then sequence 4 with 1, 2 and 3, and this will goes on and on until all the sequences will align one by one. That is known as progressive alignment method. So after that we have iterative refinement method. It is another type of multiple sequence alignment that helps to align more than two sequences to reduce the error, and it is considered as more accurate as compared to progressive method. So it works similarly as we already discussed about progressive method, but in this one, the iteration will repeat many times, as because of the repetition of iterations, there will be less error chances. Barton and Sternberg formulated this method for multiple sequence alignment. Common iterative methods are used in D-align, die-align, muscle, and probe con softwares. However, progressive refinement method was used in cluster W. So here we have iterative refinement method. It is similar to progressive alignment method, but in this case, once or new sequence is added, Once the new sequence will add, the algorithm will start the alignment from the first step and repeat all the steps one by one again to refine the results. So here we have the example that if we have sequence one, then it just aligns sequence one with two. After that, step two, sequence one, two, three. Then step three, sequence one, two, three, four. However, there is one gap or one G or one C again came in sequence three that is going to align with sequence four. Then this will refine it from the step one again. It will again start alignment from the first step. It will take more time. However, the error chance will be very less because the word iterative itself defines that the iteration will increase every time. And with the increase of iteration and with the iterative refinement method, there will be very less error chance. So it will take more time. However, progressive refinement method is also a good one and used in cluster W, that is widely used software. It takes less time as compared to iterative refinement method. However, that one is also accurate one and just give a very good results about for multiple sequence alignment. So I will perform some a small experiment for with the help of cluster W tool, so we can learn more about how we can perform cluster W on how we can perform multiple sequence alignment with the help of cluster W. Thank you so much.